So Nikhil is a researcher and professor of biology at VM. He has published a number of textbooks and is currently studying the effects of climate change on ants found in eastern deciduous forests. Erin Ellison is a senior research fellow in ecology at the Harvard Forest and adjunct research professor of biology and environmental conservation at the University of Massachusetts at Amherst. And he was recognized by the National Science Foundation and elected as a fellow of the Ecological Society of America.
So who is who?
I'm Aaron. That's Nick.
So thank you guys very much.
Sure, our pleasure. Thanks for having us here. It's a pleasure to come and be able to speak with you about ants. And so what we want to do in this talk is to describe for you a little bit of the natural history of ants. We'll talk to you a little bit about this book that will show you how to identify ants. And then finally, we want to actually take you under the hood, if the technology will cooperate, and let you see up close what ants look like, and even lead you through a few of the steps on the identification. Okay, well, what do most people know about ants? The way people usually think about ants is this sort of figure. So in the lower left, we have the queen of a species of carpenter ant that lives here in Vermont. We know this is a queen because it will be quite a bit larger than the other workers in its colony. It also has these big linebacker shoulders, which I will explain in just a second. Yes, there is damage to this wood. It is oftentimes blamed unfairly on the carpenter ants. They are associated with this damage, but they will only show up if the wood is already moist and starting to rot. They are not the cause of the problem, they are the symptom of it. That's the extent of most people's knowledge about ants here in New England, but this is only one of approximately 132 species of ants that are known from this region of the world. And we'll talk a little bit about what those ants do and what their interactions are outside of our own basements. Well, for those who grew up at a certain time, I see a few nodding heads, our introduction to ants when we were younger were these ant farms that you could buy through the mail. You would get this plastic chamber that you would set up with dirt, and you would get a small handful of ants to start with that would immediately begin building. And since their chambers are, you can see through there, you can watch all of their activities. The actual ant farm wasn't very expensive, but the marketer of this made all his money because these ants weren't going to survive very long. They don't come with the queen. So you had to send back multiple times for additional ants to keep your ant farm going. If you've ever gotten a chance to watch ants up close, especially in an ant farm, it is pretty fascinating. And we can spend our time just watching ants for a variety of reasons. I think part of the reason that they are so interesting to us is their behavior seems to embody many of the things that we think are most noteworthy and laudable about human behavior. They cooperate with one another. They work towards common goals. They make sacrifices for the benefit of the group. They're very good about all the joint child rearing that needs to take place for this entire colony. So we like to imagine that we can see some aspects of our own lives in ants. However, when we start to look a little bit at the details, we'll find that there are some things about ants that are really quite strange and foreign to us. And so I'm going to spend a few minutes for you talking about the life cycle of an ant. So here's the drawing. We're going to start in the upper left-hand corner with this reproductive queen. Now these reproductive males and females are only seen at certain times of the year, starting about now in the spring when we get warm days. Some of these ant species will rise up in very impressive mating clouds. And notice that these reproductives have wings. They are ants, but they do have wings at this phase in their life. So they will mate in this large swarm. For some species, there's only a mating with a single male. In other species, they will mate with multiple males. But however many there are, this is the only mating event that will take place for this female during the course of her life. What she's now going to do is to drop to the ground. She will shed those wings and look for a small place to burrow in and begin to develop her colony. Most will never make it. Probably 99% of these are going to get eaten by birds and other organisms or land on parking lots or other places where they're not going to be able to dig in. Some homeowners know about these. They get alarmed every spring because they think they're termites. They're not termites, but they do have this flying reproductive phase. So the queen will shed her wings. She will lay a first batch of eggs. I told you they have these linebacker shoulders compared to the standard workers. That's all that muscle tissue that's needed to operate those wings. But once she drops them and starts her own colony, she'll actually turn some of that muscle tissue to kind of a paste that will be used to feed those first offspring. Now those offspring will hatch from eggs to larvae to pupae, just like many other kinds of insects, like say a butterfly would go through this cycle with the larval stage corresponding to a caterpillar. And then from that, the next group of organisms will emerge, and these for the most part will be these sterile female worker ants. Whenever you see an ant in your kitchen or out on the ground, it is almost certainly a female. You will not see a male take on these roles. All of the work that's done in the ant colony is done by females. And as I said, they are sterile, so they're not going to be reproducing themselves. All of their effort and energy is going towards the colony. All of it is going towards helping this single queen to produce more sisters. That's what they will do. Now, this queen can live anywhere from 1 year up to, for some species, as much as 10 or 15 years. She will spend her entire life underground. As I told you, she has only that one single mating that occurs during the mating flight. She will have stored sperm from that mating, and she will use that sperm to create eggs for all the subsequent generations. So these sterile workers carry out many functions in the colony. They will take care of the larvae and pupae. They will bring food in for the female. They will construct and maintain the nest. If the nest is attacked by other organisms, they will rush out and defend it. The queen is pretty much just an egg-laying machine. She is not going to be doing anything else except producing these eggs. Now, the colony will continue to grow in size until it gets to the point where it's large enough that reproduction can begin for the colony itself. And at that point, she's going to produce a batch of special females with wings and also special males with wings. These are the reproductive stages in the life history of the colony. But the work itself of the colony, the basic maintenance, is all carried out by these sterile female workers. As I said, the queen herself can live a very long time. The lifespan of the workers is very short, perhaps on the order of days to weeks. As they die in the line of service, there are all sorts of dangers outside of the nest that they will have to contend to. And the queen is in control of this. She will make more workers as needed. In fact, the whole system is so closely interrelated, it's almost best not to think of the workers as separate individuals. It's better to think of them like leaves on a tree. So they are the functional units of this colony that carry out its specialized activities. And it's not too far-fetched to think of the entire colony as a type of superorganism, with the queen forming both the brain and the reproductive center of this. Now, as I said, there are specialized females that carry out all these different activities. Some ants even have special castes or different kinds of female workers that look differently. So in some of our carpenter ants, for example, we'll have a special group of workers that are called soldiers, that have much larger heads and jaws that primarily will defend the colony, and the other minor or smaller workers are the ones that will do the feeding. The males are only produced at one time of year. They are called drones, and there's a reason for that. They are lazy. They do nothing to contribute to this colony. They are only produced at the time that the colony is ready to reproduce, and they will usually die shortly thereafter. So as I said, in spite of the superficial appearances, there are some very different things about an ant colony. One interesting puzzle is that this arrangement is almost an evolutionary paradox. Why should an individual female, all of these workers, give up completely her own reproductive effort? Why should she sacrifice that for the sake of the colony? standard evolutionary theory, that's very difficult to predict. But you have an answer for me.
I know the answer.
You do know the answer.
Yes.
Your answer?
Because the males are, I don't remember how the mathematics works out, but because females, the sterile female workers are more genetically related to their siblings than they would be to their own offspring.
Excellent. That is the correct answer. Let me step through it and give you a few details. The first thing is that ants, along with other hymenoptera, have a very strange genetic system. It's called haplodiploidy. So here's what happens. If this female queen fertilizes one of these eggs using the stored sperm, it will develop into a female. But she also has the option of laying an egg without fertilizing it. And if she does that, that will develop into a male. The consequences of this are that females are more closely related to their sisters than they would be to their own offspring. And so it is in their genetic benefit to assist their mother to cooperate in producing more sisters. This is their strategy for spreading their genes, as opposed to reproducing actually themselves. Now, not all hymenoptera engage in this, and we do have a couple examples in the rest of the animal kingdom of other organisms that do this, that do not have this haplodiploid system. So naked mole rats and also termites have this kind of social structure, but they don't have the sexual system associated with it. So your answer is right on the money. It's the fact that the sisters are more closely related to one another that makes it in their benefit to cooperate and assist the mom. Okay, let me turn it over to Aaron, shift gears a little bit, and have him tell you something about this book.
So Nick has pointed out some of the really fascinating things about ants and about their life cycle and about their social behavior. Part of what we are interested in doing is not only teaching people about how ants work and why ants are cool, but also providing people with opportunities to learn about ants for themselves and to get out in the fields, in the woods, and the wetlands, and learn about the ants that we have here in New England. And so, as part of that, we wrote a book. We wrote the first field guide anywhere to ants. And so, you know, part of what we do is try and introduce the opportunities for people to learn about ants and learn how to identify the different kinds of ants that we have in New England, in Vermont. We'll talk a little bit about this handout as we go through to try and encourage you to get out in the field and look at ants and learn about ants and collect ants. And we, of course, will be happy to sell you and sign a book for you at the end of the evening. So the book came out about 1.5 years ago, and it was the product of it's a joint group of a joint effort, much like an ant colony, except we're not related to each other. We're not sisters, as it were. So Nick, of course, is the data geek behind the project. And so he assimilated a lot of the data. He also got out in the field a little bit, although he gets lost when he's out in the field. And Gary Alpert at the Museum of Comparative Zoology in Cambridge, who actually worked in pest control at Harvard University after he got his PhD with Ed Wilson about 30 years ago. But he actually studies a lot of the different insects that live in ant colonies with ants. And there are thousands of different kinds of beetles and flies and butterflies and moths that live with ants in clever ways. Elizabeth Farnsworth at New England Wildflower Society did all the drawings in the book. It's a field guide. We've got line drawings, we've got photographs, we've got species pages, we've got all the sorts of things that you'd expect in a field guide. She also wrote one of the chapters on the general environment here in New England. I just suck ants, or I try and herd the crew around, as it were. It really, we got it to this point. Nick will talk in a second about how we came to this book. But the four of us are all ecologists. We're not taxonomists, we're not systematists, we're not people who work in museums. We're people who are out in the field looking at ants and trying to know what they are. And so we consult all the time with people who really know what ants are and how you name them and how you describe them, and who write the really technical literature that you really don't want to have to get into in order to understand how to identify ants. It's really much easier once someone else has assimilated that for us. And so, of course, there were a whole bunch of people involved in collaborating with us, and many of these systematists. Anyone in here a systematist or related to a systematist or know a systematist? So I won't insult anyone when I say this, right? So people who work in museums, many people who work in museums naming and describing new species have the taxonomist gene, which is that they want to get everything perfect before they write it down. Of course, they're never going to get it perfect, because there's always one more ant to collect, there's one more bird to find, there's one more specimen to describe, so they never write it down. So one of the things we did was we went and talked to all of these, unfortunately mostly men. A couple of women, but they're mostly men. There's probably a reason for that, right? We go back to our life cycle. And we sort of did got them to do a brain dump for us so that we could then transmit their knowledge for this. But, so it took a lot of us to do this, but I do want to point out, as we sort of show some of these pictures of ants, some of the really interesting, weird, and natural history about ants. So this is the Allegheny mound ant. Any of you seen mounds like this in Vermont? This ant occurs here, right? These are big mounds, like this big.
Don't have a picnic on that one.
No, they are assertive. They bite, they don't sting. They don't have stingers.
They go to plant leg.
But they will bite, they will annoy you. But you know, someone asked me the other day, I was giving a talk about ants in Lewiston, Maine on Saturday, and one of the people in the audience at the end, they said, we were talking about the European fire ant, or the ruby ant, which is in Maine, and there's actually one occurrence of it in Vermont now, down south. And she said, why do these ants sting when you get near them? And I looked and I said, you know, if someone 500 times your size sat down on your roof or broke down your window, what would you do? You'd bite it, you'd shoot it, you'd sting it, you'd try to get it away. This is exactly what these things are doing. So Nick described, so this is the Allegheny mound ant. It's a beautiful ant. It's very easy to identify because its head is nicely concave, so if you pick it up and look at it, it's got this dent in its head. It's very easy to identify, but it's got a very weird life cycle. Many of our ants here in New England are what we call social parasites. So Nick described the very nice universe of a happy queen and a happy male, and they mate, and the queen falls down, and she finds a little crevice and starts her little brood and dissolves some of her wings to feed her first offspring, and all this is very nice. A lot of ants don't do that. This is one of them. Okay, this ant, when the queen falls down, the first thing she does is she looks for the nest of another ant called Formica subsericea, a little black ant. And we'll show you one of these in a few minutes. And she races down into the colony of Formica subsericea. She finds the queen and she kills her. And ants don't see very well, but they smell really well. They smell chemicals. So she takes this queen that she's just killed, she grinds it up and breaks it up and covers herself with queen juice. And so that she's masquerading as the queen of this colony. So the rest of the workers think, oh, well, this is just our queen. Yeah, she's red, but we don't see colors. It's okay. She smells right.
Like the early episodes of The Walking Dead.
Exactly.
And so then she starts laying eggs, and the host workers think, well, these are just our sisters, right? But they're not. They're like cuckoos in a nest. And so eventually, as the host workers die off, the parasite workers take over, and they enlarge this nest, and then you get one of these. About 30% of our ant species here in New England have that kind of life cycle, where they're social parasites. So it's not all one happy family.
All right, so a little bit on how Aaron and I actually got started on ants. In former lives, Aaron and I were both marine ecologists working on different things, and when Aaron started at Mount Holyoke College, And I started at University of Vermont. We kind of looked for a transition to a, well, maybe semi-aquatic system. And at that time, we started working on this remarkable organism, the northern pitcher plant, Sarracenia purpurea. This is common in bogs throughout New England. As the plant grows, the leaf folds up and forms a simple tube that fills with water, and that's a trap for insects that it captures and eats. And so we opened up our first pitcher plant to see what was inside, and there were ants in there. So we had to find out what those ants were, and that got us started. The first ant we found is actually one that had not really been recorded very much in New England at all. We later discovered that this ant, Myrmica lobifrons, is actually very common, but it's a specialist in these bogs, and so that's where you're going to find them. Anyways, at that time, it was very hard to identify ants. Both of us took a number of trips to the Museum of Comparative Zoology and sat and took extensive handwritten notes as we were tutored by a couple of the people there on how to identify things. And so for years, that's what we had to identify ants with, were our handwritten notes. And as Aaron said, part of that goal was to get that information down in a more permanent form. So the reason for writing the book, I guess, is ultimately selfish. We wrote this book because we really wanted and needed this, and we really wished we'd had it when we first began. So, people always want to know, well, why are ants important? Why should I be concerned with them other than the fact that they're incredibly cool and aesthetically beautiful? The first answer is they make and move soil. It is not earthworms that are the prime soil movers, especially here in New England. It's ants through their daily activity of moving soil up and down through the column, just their daily cleaning and maintenance of their colonies, which are underground. Remember, these ants every year in this part of the world have to get below the frost line, so that colony is going to have to move down at least a good 3 feet in most places to make it through the winter. So there's a constant movement of soil, and the soil profiles, especially in this part of the country, are very much affected by ant behavior. Also, ants are the primary scavengers in the forest. They pick up all sorts of dead plant and animal material, move it around, start to decompose it, start to break it down. If you were to pick one group of organisms and remove it all from the surface of the earth, and you did that with ants, those are the biggest changes we would see. We would really see huge differences in the way our forests look, in the way our ecosystems behave without ants there. Perhaps because of their importance, ants are the dominant fraction of biomass in many places of the world. So imagine a giant scale with 2 pans on it, and on one pan we put all the ants that we find in an area, and on the other pan we put all the other animals, vertebrates, invertebrates, other insects. We pile them up. The ant pan is going to tip lower. There's more biomass of ants in most places than the sum of all other animals combined. That's a good indication of their importance. Back to Aaron.
You can keep that clicker and let you move the slide. So the other thing that ants do in our region, especially here in Vermont, where the soils are really nice, you're all familiar, I'm sure, with the really beautiful spring woodland flowers that we have here this time of year, the spring ephemeral, so the trillium, the bloodroot, the hepatica. All of these are ant dispersed. And in fact, about 30 to 40% of our understory forest herbs are dispersed by ants. And they do 2 things here. The first is they get the seeds away from the parent plant, so that they're not going to, the seedlings are not going to compete with the parent plant. And the second is, because the ants have moved all this nice mineral-rich soil up to the top, they drop the seed in a really nice seed bed, soil bed, for the plant to grow in, for the seedling to grow in. But why do they take these seeds somewhere else? And the answer comes about through the coevolutionary interaction between the plants and the ants. So the plants need to get their seeds somewhere else, and the ants need to eat. Plants don't want their seeds to be eaten. They just want their seeds to be carried away. So all of these ant-dispersed plants have evolved to make this little structure on the seed, which we call an elaiosome, which is botanical language for a Big Mac for an ant. Because what this is, it's this glob of fats and sugars that is really attractive to an ant. They don't want the whole wheat seed, they want the junk food. And so the ants come and they're attracted to this sugar and fat mix, and if you want to go out and collect ants, you go to a picnic, they're going to go right for your cookies, right? So you can put down some cookies or a little bit of tuna fish, something with fats and sugars, and they'll come and get that. You can put down seeds, this is a bloodroot seed, with an elaiosome on it, and the ants will come and get that. They haul this back to their nest, they rip off the elaiosome, and they leave the seed. They throw that away into the waste pile. But then they go for the big map. That's the next one.
So part of this project, a phenomenal effort on Aaron's part, was to pretty much scour every museum and private collection of ants that he could get his hands on, and compile and organize all these data to tell us something about ants in New England and also about ants in Vermont. I guess I'd like to start a little bit larger at the global scale. We have about 12,000 species of ants that have names on them. We estimate there's probably another 10 to 12,000 that don't have names yet that are out there. That's pretty typical for a lot of invertebrate groups. We may, in some places, actually be losing biodiversity faster than we can even put names on it. That's how much is out there. Here in the US, about 800 species of ants. Here in New England, about 132 that are described in the book, and we estimate that there's probably at least another 20 out there that are lurking. And then for this talk, Aaron went ahead and compiled all the records that we have within the state of Vermont. And some of these records, the earliest in these collections goes back to about the 1860s, and the most recent is in 2011 when we finally had to tell Aaron to just stop, because we really did have to finish the book.
Actually, this map goes through last week.
So this map is organized not by the traditional counties, but by the older unit in New England, which are the townships. So we have the township records, and that's useful because for the older data where we don't have GPS recordings, we can at least put them on this map. And so this map shows different colors, and we have two panels here on this map. The left panel is showing you the number of ant specimens, that is the number of individual ants that have been collected in different counties in Vermont, and the darker color, different townships. And the darker colors are the higher numbers, and it gets lighter towards the gray, which is 1, and the white is no records. So we don't have any ants from those locations in the state.
Any of you live in any of those towns or near any of those towns?
So
You know someone who does?
that's on the left, number of specimens. On the right is the same structure of the graph, but now we're showing the number of species that have been recorded. So I'm making a distinction here between the number of individual ants that we collect and the number of species that we actually find. What do you notice if you look back and forth between these two graphs?
There's a lot missing in the mountains.
There are a lot missing in the mountains. What do you see about the dark squares? Where do they tend to show up?
Very,
The rivers.
Pardon?
The rivers?
Rivers, in part, but mostly what you notice here for the dark squares of species richness,
the oldest.
they tend to be where we have the dark squares of the counts. So in other words, it's not the case that there are necessarily more species of ants right around Burlington. It's just that we have collected more ants around Burlington. So my point here is that the state is largely uncollected. There is still an awful lot that we don't know about the ecology and just the basic diversity of where ants are. And we would contrast this with groups such as mammals or birds or even wildflowers and even a few charismatic other invertebrates like dragonflies or freshwater mussels, where we have very detailed maps. We actually have very good records of where those things occur. But for ants, one of arguably our most important animals in eastern forests, we still have very little information. So although there is a lot of data here that's collected from our point of view, there's still a great deal of additional work that can be done. And one of the nice things about ants is it's pretty easy to get involved because ants are easy to collect. It does not take a lot of complicated equipment. And so here's a group working with Aaron on one of his teaching workshops who are out collecting ants. And here is a typical set of tools that ant collectors use, and I'll point out a few of these things here. One of the nicest things that's happened for collection of scientific data on biodiversity is the availability of GPS. So we can actually get now not just the township or the city, but the exact coordinates. We know precisely where on the planet Earth this particular specimen was collected. Cookies are employed in a professional way. We actually use pecan sandies, and the reason is that they have both oil and sugar in them. Those are the proteins and the carbohydrates that ants seek, and they're a lot less messy to work with in the field than raw tuna fish. So that's one of the ways we can catch ants, is to put out this kind of bait and patiently wait. We have hand lenses, we have notebooks for recording information, we have trowels for digging in, we have vials for collecting the ants. We also have here a bottle of tequila in this case. Now, it is true that there is quite a drinking tradition among some circles in the entomology world, but the real reason we have this is that the best way to preserve and work with ants is they need to be killed and preserved in, ideally in 95% ethanol, alcohol. Well, that can only be gotten with a permit, and if you're traveling anywhere else in the world, you can't get 95% alcohol. So the next best thing to use is good quality, strong spirits. 190 proof Everclear is actually the very best thing, but good quality vodka or gin or tequila will do the trick. There are ant specimens in the Russian Museum in Moscow that are still preserved in the original vodka in which they were taken. So if anyone's traveled overseas to collect ants, yes, you will have a bottle of strong spirits with you.
Ants are also really beautiful to look at. In a few minutes, when we wrap up the talking portion, we're going to show you some of give you the opportunity to look at some of the ants we brought with us. We're going to project some with a portable scope that we have, and give you a chance to just really appreciate the differences in color and texture and hairiness and all sorts of things about ants. There are a couple things I'll point out in these sets of drawings. One is that these are the carpenter ants on this side that we have. We have 9 species of carpenter ants in New England. In the book, of course, we show them all in contrast with different sizes and different colors so that one can help identify them. So there are 9 species. These are the species of laziest, the so-called fuzzy ants, because they're fuzzy and hairy. We actually have 17 species of those here in New England. They come with really interesting sculpturing. Some of them have really wild heads and hairs. So this is in a group of ants. Their Latin name is Paramica. It's being renamed again, but that's all right. It's another story. We call it the Lady Gaga ant. Okay, so the real important question here is, does anyone in this room know Lady Gaga, follow Lady Gaga on Twitter, or know someone who knows Lady Gaga? I have to ask this question every time we do this.
See, we argued about this. I told Aaron that cultural reference was going to quickly become obscure.
How many people here know who Lady Gaga is? Come on. There you go. See?
It's not going to last, Aaron.
It is going to last. I tell you, if we got Lady Gaga to endorse this book,
okay, that's a
name an
different story.
advert. You know, we got Ed Wilson to endorse the book. Yeah, Ed Wilson will endorse anything. He'll write a blurb or anything. We all have Harvard connections. It's fine. What we really wanted was Lady Gaga to say, this is the best book. We contacted her publicist. She didn't respond.
You know, I get Sigourney Weaver because it's far closer to the aliens.
Ah, yes, well.
That's good.
I
Do you
mean,
think,
that
do you think
face
she'd answer
is
her email?
Definitely. I mean, and Sigourney Weaver is seriously cool.
Well, she is, but she wasn't the alien.
No, but she gave birth to one, so.
Well, you know,
Sort of like a queen.
the artist who
Science.
built that, H.R. Giger, just passed away last week. And when you look at the body structures that are in that alien, he clearly has a great deal of biological knowledge. There is an aversible pharynx, there is an articulated tail. He has borrowed parts from all through the animal kingdom.
But look, man, our goal here is to sell books. Lady Gaga endorsement is going to do better with a certain demographic than Sigourney Weaver, even though Sigourney Weaver is cool.
Please, what's the overlap on that?
We're gonna have some time at the end of this to work through how we identify ants. I just want to point out a couple of the really awesome features of the book. The book, of course, has regular keys, but it also has this really excellent pictorial key, this graphical key to ants that you can use with a 10x hand lens. And we have 10x hand lens, so you can try. This was developed for 4th graders, and it works for 4th graders, so it'll work for you too. You can take this, of course, so even if you don't buy a copy of the book, you can have the key to
Actually,
the
we should
generator.
hand these out, because that way.
We'll
we
hand
will-
these out, you can hand
Send
these out
these
so
out.
that we'll have these to look at. And we provide comparative matrix keys for closely related things that are similar that present a whole bunch of different characteristics all in one place. And of course, just like in a bird book, every species gets its own page with arrows and diagrams and nests and all sorts of distinguishing features. And where it's been found, these maps are at the county level. And as you can see by looking at this map, when you map things at the county level, you obscure a lot of the fine detail of what's going on at the town level. I would add from what Nick said, when we put the data together for the book, we had about 30,000 specimen records for all of New England that go into these maps. Only 1,700 of those records are from Vermont. Right? So Vermont not only is half the state uncollected at all, and most of the state we have fewer than two or three specimens from any town. But as a state overall in New England, other than Rhode Island, it's the most poorly collected state in New England. So we'd love it for you to get out there and collect ants and send us more information. And finally, if you do collect ants, Nick pointed out the value of GPS, so it's really important for ants to be valuable for scientists into the future and people who are studying how species might respond to climate change or to changes in land use, that they be preserved and maintained in a way that they'll be valuable for the future. So ants normally, we collect them in the field into alcohol, so they're killed quickly, and we just collect workers. We don't collect queens, so we don't kill the colony. When we get them home, we dry them out so that we can see those pretty hairs and nice sculpturing. And we glue them onto little tiny pins. And we have some examples of this to show you so you can see how to do that. And we add some labels to them so that we know where to find them. And then we put them in boxes in drawers to keep them for a long time. And this is an example. These are not all ants, right? You all recognize that this is a fly, right? But most of these are ants. And this is actually the first collection of ants from Nantucket Island off the south coast of Massachusetts. These were all collected around 1916 to 1917. They're still there. They're all nicely labeled. All the names are wrong. Because they change over time. So we actually went through, looked at these specimens, added new labels to them with current names and who updated those, put them back in the drawer at the Mariah Mitchell Museum in Nantucket. So in 100 years, someone will be able to look at them again. But so specimens that are 100 years old, 150 years old, 200 years old, just like herbarium specimens, are still usable and still valuable.
That being said, I think it is still challenging and hard for people to get into the identification of things. It seems like there's so much detail that has to be learned in order to do this. But actually, this is a skill that we all have, and I think the best way to compare this skill is to point out that most of you are pretty good at recognizing certain kinds of cars that you pass on the road. So you'll recognize, a Volkswagen versus a BMW versus a Mini Cooper. Now, if you're into cars, you know all sorts of details about those. But even if you're not, you can still recognize a couple of the main features that distinguish these kinds of vehicles. Although you might be very hard-pressed if you were actually forced to write down precisely what the difference is between them. That could be very challenging. And that's actually the challenging part of writing a key, is trying to focus in on what are the details that we use. Now, in evolutionary terms, there are certain characters that have evolved or changed with different lineages, and those characters would reveal the past history of that group. But for the purposes of keying things out, that's not actually what we want. We want to rely on characters that are a little bit more reliable, that are a little bit easy to use. So we've been talking about the genus Camponotus, and we have a couple of them up here, and they're characterized by this Volkswagen kind of profile. Now these are all different species, but they're all in the same genus, and we can recognize that genus just by that profile. Let me show you one other group. These are not in the same species, but, I mean not in the same genus, but in the same group. So this one, and this one, and this one. Why are they all together, or what groups them all together? Just looking at what's here on the screen. So that one, and that one.
Bulbous butt.
They all have bulbous butts.
No, well, that's a good point. When you first look at ants, that is actually what you first notice. But it turns out that's not a good character because the size of the abdomen of an ant often depends on when it had its last meal. So the size of the ants, even though that's the kind of character we use, is not always a good character. Sometimes color's not always a good character because that can vary and it can also change after they've been preserved. So instead we look for structures on these ants. Back here,
Would you point out the ones again? You want us to try to?
that one, this one, this one.
Between the thorax and abdomen, what's going on?
Exactly. What do you notice here that's common? What's happening?
Looks different. It's all small and.
So what we have, anyone else? You're on the right track. What is pulling out those three ants from all the rest?
Are those actually three segments, or are those two between the abdomen and the thorax?
Yeah.
Two little bulbs right there.
Right, so this,
Segments,
yep, there we go. This structure is called actually the petiole and the postpetiole. Now, of course, these are insects, which means, yes, there is a head, thorax, and an abdomen. But in the ants, just to confuse things, a couple of the first abdominal segments have been fused to form this little intermediate structure that we call the petiole. And all of these ants have a petiole with two pieces to it. Other ants, like these, have only one element in the petiole. So this is just an example of the kinds of characters that we use to look at different ants and tell them apart. And we'll see some of these, I hope, here in just a second.
I will add, all the terms are on this side. All right, so this is the back flyleaf of the book. This is the front flyleaf. The key thing, the reason that we have these up here, these are the most common, 5 most common ants in Vermont, and the 5 rarest ants in Vermont. When you look at an ant, when you pick up an ant to look at, almost the genus, we have 32 genera of ants in New England, 31 genera of ants in New England. All but 3 can be identified just by looking at them in profile. So it's just like when that VW goes by and you see that logo on the back and you say, that was a VW. With ants, you look at them from the side, 28 of the 31 genera you get just by looking at the side. You don't have to go any further, right? The other 3, you have to look at the head and the teeth. But there's enough on the other sides to get you there too. So simply seeing them from the side is like looking at cars you drive by. And you can do that with simple hand lens.
So our point is that this is not as difficult of a task as it may seem. It's just a little unfamiliar in the beginning because these structures do look very different. If you keyed out birds or any other kind of group of organisms, this is, I actually think, easier than some groups because these structures are pretty well defined and pretty easy once you start looking at them. There's still some challenges, but not that.
How many of you do go birding? Right? So you all have binoculars, right? So apparently, I don't birds, I don't know this sort of thing, but apparently if you turn your binoculars around, you can use them like a hand lens.
Absolutely.
Yeah, so you can do that, and you do your ants. You don't even need an extra device. But if you can do all the birds in New England, you can do all the ants in New England. You can do all the ferns in New England, same number of ants.
Okay, so I want to talk just a minute about ants as an entree to studying biodiversity. And before I do that, I would like to make the argument to you that it is actually ants that are ultimately responsible for today's environmental movement. Ants. And this story begins at the end of World War II with the development of synthetic pesticides and fertilizers, particularly the development of DDT. And not too long before this, a particularly nasty species of ant showed up in the southeastern US, the red fire ant. Anyone here traveled in the southeast, had a run-in with the fire ant? You want to tell us what that was like, anyone? What's your fire ant? Everyone has a fire ant story. What's your fire ant story?
Producing a video on golf instruction on a course, and the cameraman stepped, and he had to get naked fast.
Fire ants are unbelievable. Once you have encountered them once, you'll never forget it. Anyways, for all sorts of reasons, they're a huge problem with agriculture. They like disturbed areas like suburban lawns, so they're right around families. With the development of DDT, a massive effort was undertaken throughout the southeast to exterminate the fire ant. And thousands of acres were literally coated in this. All this was going on at the same time that an overseas adventure was taking place with an herbicide called Agent Orange. And unfortunately, both of these efforts on our government's behalf ended up as complete failures. The fire ant was not exterminated from the southeast, but huge quantities of DDT were released into the environment. Not too long after that, Rachel Carson's book, Silent Spring, appears. Not too long after that, the first Earth Day happens in this country. So I'm going to attribute our current environmental awareness ultimately back to ants, ultimately back to this one ant.
Do they have the chitin structure of termites and cockroaches so that they can withstand radiation levels as well?
Yes, ants are particularly good, and there are some great, what's the science fiction movie, Them? It's about the ants, the 1950s movie about ants that are radiated and grow to giant sizes. But yes, they are. Like many insects, of course, they take in air through spiracles on their sides, and when they're exposed to a pesticide or something else, they can just shut those closed for a period of time. So, say a few things about ants and backyard biodiversity. We tend to make a very strong distinction between the human world and the natural world. When I say we, I mean both scientists and the general public. In fact, we scientists are probably worse at this. We spend most of our careers trying to work in places that appear to have little or no human influence. And so we make this big distinction. But in fact, the boundaries are rather blurry. One of the things we've learned from ecology over the past several decades is that there's probably no place on Earth that has not been affected by human activities. Both through climate change, through our use of resources, even these apparently pristine places, including the bogs that Aaron and I love to work in, are affected by human activities. But the converse is also true. There is no such thing as a completely barren urban environment. Our modification of the environment actually creates niches or habitats that organisms use. And ants, in particular, are oftentimes associated with humans. And so starting to look, you don't go to a pristine forest and we say, well, we'll go out and look for ants. Try looking in your own backyard, which in some cases is the very best place for looking at biodiversity. Particularly because many ants are thermophiles. They love warm little microclimates that we create through our own activities. Many years ago, I was on a collecting trip for ants in the Smoky Mountains, and we went up and down the hillsides and laid out transects in beautiful old-growth forests, and we did pick up a number of species that live in those habitats. But if you really want to get ants in the Smokies, you go to parking lots and campgrounds. Those little warm patches, you'll pick up all sorts of things that are attractive to them. So we can go ahead and actually look at the biodiversity in a backyard. And so what I did for tonight, this is the first time Aaron and I are going to try this, is I bought some ants from home. So last summer, I walked around my backyard. I live in Burlington, Vermont. We're up in the Hill District. My wife is quite a gardener, so we have lots of perennial plants growing in our yard, but it's not an especially large yard. And what I did was to walk around the yard over a period of maybe 10 minutes on a warm August day and simply take these little vials, this is 95% ethanol, what we use, and look for ants. Everyone has a different way of collecting ants. I prefer to work with my fingers. I find that is the easiest way to catch them and pick them up. And here in New England, we really don't have anything that's going to sting or bite you. Maybe the ruby fire ant that Aaron mentioned if you're coastal Maine or some places, but even then you're not going to have to worry much. So the ants get picked up, I just dropped them into the vial, and I simply walked around in a couple of different places, including our brick driveway, which, as you will see, actually is kind of a biodiversity hotspot. And so if we can bring up the screen here, our plan is to try and show you what we've got.
It's not.
Not happening? Okay.
It may go on, it'll certainly go on my laptop. I'm gonna try plugging this in, but I don't have, your software.
While we're waiting, any questions
This is
or
on digital
things you want to know? So you're first?
From an ecological standpoint, why is it important to have numerous species of ants in a certain area?
Well, that's a good question, and I would say that's an open question. We don't know for certain whether it is in fact necessary to have lots of species, or whether perhaps one or two species will do most of the job. I would say that for plant dispersal, so a lot of these trillium and things that Aaron described, there are only particular species that will carry those eliosomes and are found in the forest. So for those kinds of activities, there are particular species that...
So what is, I'm sorry, let me just start.
Back here?
Yeah,
So what is projected?
you said they kick out the dead stuff. Does that include things like a reindeer ant, like
Yes.
deer antler?
So you have a DVD.
The
Yeah, they will-
mysterious, you can never find the deer antler once they do file off. It's the ants.
They will strip off surfaces
But it's-
of dead and decaying material of all sorts of things. So again, in the south, with fire ants, they will strip skeletons
You have a
clean.
stick that you
Some
put in
forensics
somewhere?
operations will use ants to clean skeletal material. And some ant records have even been used in forensics cases because they will show up at corpses.
I do this for,
Yeah.
so let me
And then
just get
for
clear.
some of the work you're doing,
Where
are you
is-
also using this study to test, the movement of
So we should
invasive
have-
species or more vector-borne, or not as a vector-borne, but a migratory, like what's the spatial range of
Right.
an ant? Is it
So,
a mile,
okay.
100
If I could get
feet?
it out to the back.
When you say spatial range, you mean movement of a single individual?
Yeah, like
Okay.
how far,
Sure.
I mean, up and down like 3-4 feet, but
Okay, so in terms of horizontal distance, many species move a surprisingly short distance. Ground nesting things might only move a few centimeters or a few feet. Some of your larger carpenter ants will go many yards away. So at this time of year, when you have carpenter ants and wood ants coming into your house, those are oftentimes satellite colonies that have started to spread out from a main colony that might be in an old rotting stump way in the back of your yard.
Nope.
So they can move those distances. The other thing is in New England is not a proper biogeographic province. And by that I mean we don't really have species of ants that are strictly endemic to New England. What we have instead are warm southern species that come a certain way up into New England, and we have cold northern species that come down in their ranges a certain distance. So ants are going to turn out to be useful
Get
for,
the
I
menu
think,
here.
tracking the effects of climate change, because what we're going to start seeing are more and more appearances of these southern species as the climate warms up, and we may see the disappearance or the northward migration of some of these cooler climate species.
because I know in Delaware they've had invasions of fire ants,
Right.
and
So
they're thought to be like
Do you have
Georgia,
another?
it's like how they get up.
there are several ant species here in New England that only occur in greenhouses or in hospitals or other warm microclimates. But it's easy to imagine if the climate gets much warmer, they will be able to expand out from that. The fire ants and other things get a foothold because they come in on nursery plants, and it's a huge problem. That's a major vector for lots of organisms to get moved around.
So we're responsible also.
We are, yeah. Here?
So how different are the species found here in New England, say to the UK?
There are several major ones that are shared, that are the same, and then we also have others that are in the group that might be a different species, but that's in the same genus. And one of the things
So
that's
this book
interesting...
could be used in the UK?
Somewhat. You'd be much better with this book throughout other areas of North America, that would be the better place. But you could go quite a ways west and north with this book, and even down into southern forests.
Okay.
One of the things that's interesting here is in New England, we don't have that many introduced species. I think only about 10% or so. Whereas in Florida, you've got 25 or 30% introduced species, most of which, as you might guess, are tropical. And one of the things I wonder about is that this part of the country, we've had so much contact with Europe for such a long time, some of the things that we are calling native ants to New England may, as Aaron mentioned, we make a big, yeah, in here also. We make a big distinction between collecting workers, which I am comfortable doing, versus collecting queens, which kills a whole colony. And the problem, though, is with ants, you really do, if you're going to get them down to the species level, you are going to have to kill them and pickle them in alcohol. You really cannot, for the most part, identify them to species. But with your hand lens, you can get pretty far. You can get them down to the level of genus easily. Some of them you may be able to do in the species.
With practice.
With practice.
You can do it, the species with a hand lens, at least with a 20x hand lens, with lights.
So one of the useful hints you'll see in this book is the book instructs you that if you want to look at ants, once you've collected them, you have to dry them out. And I made sure that that ended up in the book because my earlier work was on marine invertebrates, and when you look at marine invertebrates, you put them in a petri dish with alcohol and see their features. If you do that with ants, you cannot see anything. And the first couple weeks I spent trying to identify ants, it was impossible. I could not find the simplest character, and someone had to sit down and show me that, no, you actually want to dry them out in order to see this. So, again, like I said, this is the book we wanted. I wished someone had told me this. So, a little bit about this ant. I told you I've got these from my backyard. In our backyard, near one of the back flower beds, is this large, not that large, just about this big, maybe about this high, ant mound. That these ants are crawling around on. And I know my wife would very much like to get this unsightly mound out of her garden, but that mound has been there for over 15 years. So there is a single queen or a couple queens in there that have produced these workers, and now we're seeing the workers from, it's pretty good, it's pretty good.
And this is the species that Allegheny mound ant comes in and parasitizes.
So if you've got your key, I want to actually lead you through how we would do this just to get us down to the genus level. So let's start in the upper left-hand corner. And the first thing we need to decide is the size of the worker. So you see these little scale bars for comparison. Do we have a big ant? Do we have a medium-sized ant? Or do we have a tiny ant? And in the ant world, this one will come out to be a big ant. So that's our first spot on the key. So,
of course, you could come up and look at this ant, and you will see that it doesn't seem very big, but in ant universe, it's
in
pretty
the ant
big.
universe, this is pretty good sized. Oh, that's nice, Aaron. That's really very impressive.
It's drying out slowly.
Yeah.
But you can see there's enough detail on here now that you can determine the genus of this
Yeah.
ant.
So let's now go through this key. So what we're going to do now is we're going to move to this box in the upper left-hand corner, which says, do you need one here? Okay, you got one there? Okay. So this is group 1 ants greater than 5 millimeters in length. And now you're going to see a forked arrow at the top, which is going to take us to our first, what's called couplet, where we're going to make a decision. And what we're looking for here is the structure I mentioned before called the pedicel. And it's either got one segment to it, or it's got 2 segments. And when we showed that slide before, we were looking at ones that had 2 segments. So my question for you, for this ant, does this have 1 or 2 segments on its pedicel?
Great big one.
Yeah, this has got 1. It's pretty clear. It's right here. So that's going to now take us down to the left-hand side of this key. And we're looking at the mesosoma. That's the $5 word for the top of the back. And the choices are whether it is smoothly convex. By that we mean it makes a nice smooth arc over the top. Or whether it is lumpy. good taxonomic word, lumpy, but that's precisely the phrase that we want. So the area that we're looking at here, Aaron, I think here we go. Oh,
Yeah, no, I know what
Okay.
it is.
The area that we're looking at here is this is the part of the body that
I
we're
was
looking
looking
at.
for camponotus.
Oh, okay. Oh, there is one in here,
What I'm gonna do is I'm gonna put two
I
on
think number
pins.
five, there's number 5 in there. Anyway, so what's your choice on this couplet? Is this a smoothly convex or is it lumpy?
Lumpy.
Yeah, this is lumpy, because look at this, it's gonna go up, then down, then up, then down. So that's going to lead us to this next arrow. We're down here at mesosoma lumpy. And now it's asking us whether the mandibles are normal or the mandibles are forming a sickle shape. And that's going to be pretty hard to see here. The mandibles are here, but we're really only looking at the side of them, and they're closed. They're fairly compact, so I will just give you the answer on this one. These mandibles are of the normal or typical form. Okay?
Plus, of course, the ones with sickle-shaped mandibles are bright red, but that's another
Right.
story.
They're really quite evil looking, and you would see this saber-like structure. You probably could see it on this, even on this magnification if we had it.
But what I'm gonna do here is I'm gonna pass this around, and don't worry if it falls off, it's okay. I'm gonna pass this little set of pin dabs around. And the one on, these are both big ants. This is classified as big ants. And the one on the pin with the lumpy back, which you can see this with just even my 55-year-old eyes can see this. This one has the lumpy back, and this is Formica, and this one has the bent back, which is Camponotus, the carpenter ant. And so those two genera, big, dark ants, can be distinguished by the shape of their back alone. And you should be able to see that with the naked eye, but if you'd like a hand lens,
Why don't you send one around with that? That'd be good.
I'll send a hand lens around with this. And everyone here knows how to use a hand lens, is that right? And please tell me if you don't know how to use a hand lens, because it will make your life much easier if you admit it now. So I will tell you, for those of you who are too shy, which is you put the lens to your eye, and you bring the specimen to the lens until it snaps into focus. It will not work if you go like this. It will not work if you go like this. And I know you all know that. But if you don't know that, like this.
So back in our sheet here, you have now identified this ant to the genus level. So,
And these are very cool hand lenses, 5, 10, 15. So you can see it really cool if you go to 15.
so you're at Formica.
And for the real connoisseurs, 20 with a light. Species level.
So if you wanted to go to the next step, we have 31 species of Formica in Vermont. So you would then have to dig into the keys themselves to find out which species we have. Some of the characters that are used here have to do with the silvery sheen on the body, which is very hard to see in this ant, but if you looked at this under bright light, you would notice it. This ant has a very long scape, that is the length of the very first segment of the antenna, and there are some other features about the silver coloring that will help you pin it down. So it's really more of the same, but this key is designed to be one that you could actually use outdoors and for the most part get to the genus that you need to.
Look at it with the hand lens too. They're really cool looking.
I have another question.
Yeah.
Once the ant pupates to like its adult form, do they stop growing?
Yes.
Okay. Is that how you can determine
Yeah,
if it's big or little?
Right.
Right, yeah, that's not going to change much. Now, as I said before, the size of the abdomen will change because the abdomen is flexible integument, so it'll swell out if it's eaten and shrink up if it hasn't. But the main body parts will not really change in size.
Now, the one distinction that I will make between the handout of that and the flyleaf in the book version, that was expanded to fit the page. So the little scale bars are not the real size. So when it says 5 millimeters, it's a little bigger than 5 millimeters. But in the book, it's actually 5 millimeters. So if you hold your ant there, you actually have a ruler. We did it based on size, because the first thing that 4-year-olds say is, "That's a big red ant." And big is always first, or "That's really tiny." And so we figured that was a good division. Most of us, what we do when we look at these is we start by saying, really, does it have one segment in the middle, or does it have two segments in the middle on that pedestal? That's the first division. And that starts moving you down. We can go through them with the scope. If anyone would like to try on their own, I have, 400 pinned ones here and hand lenses, and you're welcome to look at them. They're already labeled, so you can cheat. Right? But you're welcome to come and look at them. They're pretty colored. They don't bite. They're dead.
In the collection I have here, just 10 minutes walking around my own garden, I pulled up 6 species, 3 of which were on our brick driveway from an area about this big. And I was not working hard here, so it would not surprise me if there are another 6 in my garden that I have not come across yet. That's the level of diversity you can find with really very little work or effort involved.
But just to give you a sense here, so I will put one next to the Formica.
So,
Okay, so this is a little one next to the Formica. In the key, that's a medium size ant. Okay, that's not a tiny ant. That's a medium size ant. Okay, now this one's very interesting. This one has some really excellent field characters. We're all about field characters because we're field biologists here. Okay, and so this one, I'm not going to try and turn it on its side, but you should be able to see, or you would if you came a little closer to the screen. I was realizing we have one of these screens at Harvard Forest, and we think, wow, it's really cool, it's really sharp, but it's actually really small, and so when you're in the back, it's a little hard. But you can see it's got 2 segments on the pedestal here. You ought to be able to see that. What did I do?
You touch the screen.
Was it me or it?
Oh.
No, it was my computer.
I actually doubt that that's a touchscreen.
Anyway, it has 2 segments on the petiole here, right? And it's small, but as a field character, this one, besides having the 2 segments, which is the first thing you look at, you see how the legs are like tan and then dark muscly and tan and dark muscly? This is actually the only species we have in New England that looks like this, right? And so those 2 segments and these sort of dark muscles on the legs tell you that it's Temnothorax longispinosus. So you get the species of this. Yeah, I can give you the common name. We don't have a common name for this. We made up most of the common names in the book.
It was that last step.
We made up the common names. You could call it the long spine Temnothorax. How's that? It's sort of like the red-headed flycatcher. This is the long spine
Get more than
Temnothorax.
a B plus
So I'm gonna.
I just can't.
So I know, I realize I'm all about selling books, because you don't write a book unless you're going to sell. But one of the things that we do in this book that I think should be done in bird books, should be done in plant books. We all scientists.
Is anybody actually making books now? I thought everything was going to be digital.
30% of book sales are digital, 70% are on paper, it turns out. And it's been flat for the last 4 years, which is very interesting.
Are you gonna do this as
This
an
is
app?
available as an ebook.
Okay.
If someone would like to donate $70,000, we will have an app made, but it will cost us $70,000 to make an app. So if anyone would like
Oh,
to,
we've done that before with a movie. It's about 70,000.
that's right. So if anyone would like to contribute 70 grand, we
50 to 100.
will turn it into an app. We have quotes. The folks who did the Audubon Guide app for the birds will do this for us for $70K.
Now, who told you that? Since I made that app, so I want to.
We contacted the publisher of that and said, what would it cost? And they said $70K. Yale University Press went out and got bids.
Oh, well, that's Yale University Press. Our digital company.
They own the copyright.
Is here in Woodstock, so.
Would you like to bid?
No, but I would do it for you.
Would you? We have wireframes ready to go. And we can provide all the images. Well, let's talk afterwards. That'd be great.
Yeah.
But the point I was going to make is that every genus,
He's getting this.
every genus and every species we talk about why it's named what it is, so that you can think about that. So this is the genus Temnothorax. What does Temnothorax mean? Right, so that you can remember it. It means divided. It's divided because this part of the mesosoma has a suture, right? And so it's divided. So it's a divided ant, right? And then this one is longispinosus, which means long spine. So it's the long spine divided ant. Now, I find it easier to remember Temnothorax longispinosus. But I studied Latin in high school and junior high school, and so I know how to make this translation. We do this for all of these. You know, there's Temnothorax curvispinosus. Guess what that one means?
Curve spine.
The curve, you're good, right?
I did that one.
Right? So then there's Temnothorax shomai. That's named for this guy named Shom. There are a lot of taxonomists name things for their buddies.
They do, and it's.
They do. But you learn all sorts of things. So there's Aphenogaster tridi, right? And there's Dolichoderus marii. So both of these are, there's Mary's dolichoderus, and there's Treat's Aphenogaster, both of which are named for Mary Treat, who was an entomologist and botanist working in the New Jersey Pine Barrens in the 1800s and who corresponded with Darwin. And was a really cool naturalist in the 1800s. And she's got 4 species of ants named for her because she collected the specimen and mailed it to the people who named it, and they named it for her. So you learn a bit about the natural history, the etymological history as well. Anyway, this is a medium-sized ant. Right? It's a big ant. I don't think you have any tiny ants, do you?
There should be, I thought there was a Tetramorium in there. Is that number
Tetramorium's a medium-sized ant. We need a Solenopsis.
2? Oh, no, I don't have any.
Yes, we do.
Not that Solenopsis. The Solenopsis we have here is really, really tiny, and it doesn't bite like the one down in South Carolina. This one's pretty small.
I still remember Cephalus spidomorphi, but it.
Here we go. This one's very common. This is an extremely common ant here in New England. This is Brachymyrmex dipillis.
Do you repeat that?
Brachymyrmex dipillis. So what do you think so we'll start with the easy part. Myrmex.
Brachia is easier.
Not a
Mermax.
Myrmex. Myrmex comes up a lot in ants because Myrmex means ant in Greek. Formica, this one,
Okay.
is not plastic.
Okay.
Formica is Latin for ant. So you've got ants in Latin, ants in Greek. Yes, sir.
May I ask you about ant hills?
In 1 minute, let me finish Brachymyrmex and we'll talk about ant hills.
I just-
Brachy is short.
The human interaction here is more interesting.
Yes, of course it is. Brachymyrmex is short horns because it's got very short antennae. So it's the short-horned ant, and it's depilated. And they're almost all women here, so you all know what depilated means.
You?
Some of us of a certain age may have abandoned this when we were children as part of the feminist revolution, but
Oh, without a bra.
no. What else did women used to do before the feminist revolution?
Oh, man. Wash dishes.
Get it permanent.
They used to shave and pull hair out. They depilated.
Ah.
Oh yeah, you remember this. Hairless. The hairless short-horned ant.
Okay.
What can I tell you about anthills? Anyway, I'm going to pass this one around. There are 2, one on each pin. This is a tiny ant. This is a small ant. This is 1.2 millimeter long ant. So I'm going to start it over here.
So medium man is 4 millimeters
Medium man is 3 millimeters. Don't worry about it.
on
I
average?
think 3 to 5.
3 to 5 is
Okay,
medium.
so for, I need to know because I have a climate cartoon I use in my talks, and it's a
Here,
human
take
worker
it.
versus a human ant. And I compare it to the
Take a hand
height of the
on this.
troposphere, and I've been trying, but I'm off by a factor of 2 then. It's not a 3-story building, it's a 6-story building.
Okay,
It's like that humans to the troposphere equate to a 4 millimeter standard ant is equivalent to a 6-story building.
Okay.
And if there are 7 billion of them in your house, you will notice them.
You might.
So that's my,
So what can I tell you about anthills?
So you remarked there's your brick driveway.
Yep.
So I have a turnaround, it's kind of bare, but there is some grass, but every spring, there are mounds of ants.
Yeah,
And
absolutely.
there's probably 15.
It's probably the pavement ant, Tetramorium cespetum. The Tetramorium for 4 segments, tetramorium, right?
Is that a
And it
small,
refers to,
medium,
it's a
or-
medium ant.
Medium.
And it's got 2 segments,
Mhm.
and it refers to actually the fact that the palps, the little sensitive things behind the big mandibles, have 4 segments in the type genus. Now, it turns out lots of the species have different numbers of palps, but that's okay.
So
it's
they also
tetramorium.
have what I would consider to be a large ant that has fallen in love with our toothpaste. And I find them in the sink. It's probably gross, you know, after brush your
Some of
teeth.
us wash our sinks.
If it's a
Well,
large
I'm
ant,
impressed now.
If
if
it's a large
ant
ant.
here in New England, it's probably one of the 2 that we've seen tonight. It's either this Formica up here,
Yeah.
or it's the Campodotus,
Or it's
the
a
one with the
carpenter
smooth
ant.
back.
Okay.
Anything that's
So why
large
would
enough
they-
to see.
It's probably silly. Why would they move inside? Are they outside and coming inside?
It's warmer in
And
your
they're-
house.
Will they go back out?
They will. Yep. So
And
I
you
have
need to
this
stop
argument
feeding them toothpaste.
at home every year
I
about
think
this
they
time
like,
because
I think they like the soap.
they do come in. They come in for the warmth, so it's still
Yeah.
cold outside, but the colony started to become active.
Yeah.
This looks like a good place to come in.
And the ants in the bathroom are different than the ants in the kitchen.
That's,
Yes. And in fact, so the ants in the kitchen,
See, you think I don't pay attention.
you do. The ants in the kitchen are small ants, small to medium-sized
Smaller,
ants.
yeah.
They're what we all call sugar ants, right?
yeah.
So if you grab one of these ants, easiest way, you lick your finger, touch the ant, you've got it on your
Do it in that
finger.
order.
You,
Do it in
yeah,
that
you
order.
lick your finger before you touch the end?
Before you
All
touch
right.
the ant, then you squeeze her gently. You won't hurt her, she has an exoskeleton. And you smell your fingers, and it will smell like bananas.
Really?
And this is,
Best way to know that that's the ant.
and this is Tapenoma, here's another one, this is Tapenoma sessile, okay? So Tapenoma, so Nick pointed out you've got the mesosoma and you've got the gaster, so the middle part and the back part, and you got the pedicel in the middle. And you can always see the pedicel, except in Tapenoma. The things are overlapping like this, and the pedicel is fallen. Tapenoma means fallen in Greek. Sessile, how many of you know botany?
Did you
right?
take Greek in high school too?
No, I took Latin, but I came with Greek at the end. Sessile, so what's a sessile thing in plants? It's attached. So it's a fallen petiole that's sessile. So it's the fallen sessile ant. So it's Tapenoma sessile, and it smells like bananas, and the official name from the Entomological Society of America is the odorous house ant, because it lives in your house and smells like bananas.
So,
Now, how did they get in?
How did it get in? It
Only in the spring.
So it goes in anywhere there's a crack in your foundation or your shakes or your siding, and it's following moisture and it's looking for sugar. And if you don't want it, put your sugar in a glass jar and seal it tight and they'll go
We
away.
call them water ants.
Yeah, because they follow the water.
Okay.
so can I ask you another question? So, since we generally put all creatures that we find in the house outside without stomping
Ain't
them,
gonna make any difference with that.
is there, they don't bring any pathogens in that we needed to
No,
be concerned
so
about?
having ants in your house is very different from having cockroaches, mosquitoes, ticks, bed bugs. None of these things are gonna be a problem. There's not gonna be an odor. I mean, I guess if you actually get a full-blown nest going in your woodwork, you can have some problems, but for the most part, the best thing is to just keep the counters clean. As it warms up more, they're gonna go back outside.
Like your sinks, is that?
Exactly.
Yeah, wash it up after you brush it.
I think having them in the house makes your kitchen look like something from an M.C. Escher print. So I am happy to see them crawling across surfaces, yes.
Are they attracted to fresh paint?
I haven't heard that, Aaron.
I don't know, I'll find out, I
I
gotta
mean, they
repaint my
certainly
house.
are,
Oh, you know what,
everything
the
is
Blue
different.
Jays
No,
have
the
been
new,
eating it.
the new paints, a lot of new oil-based paints are made out of vegetable oil base.
This was latex. I did 2 rooms over last year, and at 2 different times of the winter, one in like February, one in March. And each time I did, all of a sudden I had ants in that
Huh.
room.
Well,
And
it's
they left
things-
after a
little base,
little
yeah.
while, but,
Possible, sure.
They use
You got a question back here?
what was that?
They're using a little.
With the camera, did you have a question?
Did they?
Oh, I'm sorry, I thought I saw you had a question.
Who determined the odorous house scent smelled like bananas?
Well, I mean, some people say
it's
coconut.
like coconut.
No, but like,
Smell it, and you're gonna pick it up, you're gonna smell it, and say, bananas. It smells
no,
like
by all
bananas.
means I believe you. I'm
It's
just
the
wondering
esters
who decided I'm
in
gonna smell
bananas.
this ant.
I don't know, but there are other ants, so there's a
How many
couple
people-
ants that smell like citronella or lemon, and they're, you
How
can
many-
almost smell those from the colony itself.
How many people here go out and identify plants? How many scratch and sniff plants are there? The first thing you do, you take the leaf, you grind it up, you smell it. We use all our senses to identify things, all right? You can't see them. Someone knocked them off. Here, if you bring it over here, I'll project that one if you bring it over here.
No, they're very tiny.
They're very tiny.
Oh, good, I dropped it. I thought maybe it was
No, I
left
reached-
down
we
the
reached-
floor.
You dropped it, you broke it?
Judy, it's all your
I
fault.
saw, them again.
I did, well, I saw something.
Bring it up to your eye.
Are they-
It's not-
are they-
there's only a limited
It
plane
turns
where
out
the
that she
focus
dropped them,
location.
so they could have fallen off.
Okay, well, yeah, you're not gonna see much on the tiniest ones, except for the
She
size.
could have destroyed my, very
Oh,
valuable
go
specimen.
down here.
Down there, they're walking away.
Now, what about the edibility of ants? What's the,
How hungry are you?
well, I mean,
Only in
what's the
chocolate.
nutritional value? Well, the chocolate covered ant fat and all this, and bugs,
Micro
What is it?
livestock.
66% of human
There's an FAO report from last year about
Like
micro
66%
livestock.
of human beings have insects in their diet.
A couple of years ago, I took a trip to southern China in the rainforest, and one of the things they serve there, they call them ant eggs, but it's actually larvae of a weaver ant that lives high up in the trees. It tastes a bit like risotto. They serve it with hot chilies and greens. It's pretty good. To get these larvae, what they have to do is climb up the trees and dig into the nests. The ants bite and are pretty aggressive, so they climb up naked because it's easier to brush the ants off your skin if you're not wearing any clothing.
But because there's a huge nutrient source.
Right, it's
But
rich
it's the
in-
larvae, because once you get the chitin on, it's a
Right.
little harder, okay.
Exactly, so they call them eggs, but it's definitely the larvae
But
that
is,
they're
is
serving.
there a non-edible version, or is ants just in general a good, I mean, it's one of those survival foods. Locusts and ants.
I'm sure, like
Like
all
fire
sorts,
ants, they're
I'm
not
sure
edible,
just
are
like
they?
plants, there are gonna be some that are good to eat,
Okay.
some that taste nasty, and perhaps some that are even poisonous. I would expect it's exactly the same way.
Brownie
Do you add that to
There's
your
one on the bottom.
classification, edibility?
Edibility, no, we haven't done that. One thing that could be added to the classification that we mentioned in the book is, now handheld video is really cheap, and one of the characters that we would like to have had in this key is what the nest looks like, the nest entrance.
Oh.
So a great school project would be to have people videotape the nests and also the movement of the ants in and out, because that can be characteristic. Then pull a few specimens, identify them, and you can come up with a key for identifying them based on the nest entrances. I think it would be great, but it hasn't been done yet.
I have a little video
Right?
camera this big, and a little pencil, and it goes great big in the small hood.
So this is it, 100 eggs.
Okay,
See, you didn't.
I didn't drop it.
You dropped one. It's really small.
It's here.
It's really small.
So just so you know what you're seeing, that triangle is a tip of a stiff piece of cardboard, and the ant is actually glued onto the end of that tip.
What do you use for glue?
White glue.
White glue?
Some
White
people like
glue
hide
or
glue,
white
it's a
glue?
huge source of controversy. People have very strong emotions about this particular issue, what kind of glue to use.
And the reason
Well, because
is,
we,
the important thing is that it be water soluble glue.
oh, why is that?
So that when I need to take it off to do a better identification, because when you glued it, there's more glue than there is ant, then I can just dissolve the glue away.
Okay, because when they do,
Here's one that hasn't been dropped
For
on the
the
ground.
forams for benthic studies and oceanography, they use super glue.
Yeah, but that's because they're dealing with silica.
Because they've got a silica
Right.
base.
Isn't shellac also a viable option for ants?
No,
No.
it's not water soluble, it's alcohol, well, it's close, it's alcohol soluble, so that can be okay.
Does anybody want to test the binocular theory?
Oh,
You
yeah.
can test the binocular here, you can test handguns, you can take some microscopes, you're welcome to come up and play with these, you don't have to sit
Oh,
in
by
the
the
class.
That's it, we are done for our
So who
presentation.
was
So thank you.
Yes. So,
May I